Welcome and thank you for listening to my lecture on Chapter 5, which is all about energy flow in natural systems. So I'm going to start this lecture with section 5.3 about chemical reactions. There's a little bit of vocabulary that I think you may already be familiar with, but that I would like for you to know. And I wanted you to know the terms reactants and substrates and products. So if you've ever had a physical science or chemistry class, the reactants are just the molecules at the beginning of a chemical reaction, the starting molecules. And in biological systems and in biology classes in general, we call the reactants substrates. So if you hear the word substrate, you know that that is basically the same thing as reactant. And the products are the molecules that are formed through the chemical reaction, or the products that are on the right hand side of the arrow in a chemical reaction. So there are two types of chemical reactions that I'm just going to briefly talk about. Endergonic reactions, which have products that have more energy than the reactants. And in endergonic reactions, the reaction requires energy, requires an input of energy in order to go forward or in order to happen. And then the second type of reaction I wanted to bring your attention to is called an exergonic reaction. And exergonic reactions have products that have less energy than the reactants. And exergonic reactions release energy, so they don't take an energy input in order to occur. Okay, so even an exergonic reaction may require an initial input of energy, and that's known as activation energy. So that's just the energy required to get a reaction started. And the activation energy usually works to destabilize the chemical bonds within the molecules that are the reactants or the substrate. And reactions, oftentimes the activation energy necessary to make the reaction go forward is a barrier for that reaction to happen at all. So if the activation energy is lowered, then the reaction is more likely to happen. And so lowering the activation energy is a process that we call catalysis. You've probably heard the word catalyze. So to catalyze a reaction is just to lower the activation energy, which makes the reaction happen much faster. And this is figure 5.4 in your book, and it's just showing the process of catalysis, or of catalyzing a reaction. And you can look at that one in your book. Okay, so we have seen enzymes before. That was in chapter 3 when we covered macromolecules. Enzymes are specialized proteins that act as catalysts in reactions. So you can think in your head of an enzyme as a biological catalyst. And so enzymes, like all proteins, have a tertiary or three-dimensional structure. And within that structure, they have a pocket called the active site that is specifically shaped to bind its substrate. So every enzyme binds a specific set of reactants or substrates. And so we had talked about tertiary structure in proteins being important for function, and one reason that the tertiary structure of an enzyme is important is because without that specific three-dimensional structure, the active site would not properly fit the substrate, so the enzyme would not be active. And so the binding of a reactant to an enzyme causes a slight change in the shape of the enzyme, and this is known as induced fit. And so whenever the enzyme binds the substrate, they fit really tightly together and form a sort of a complex or a unit. And the enzyme, by stressing the bonds in its substrate, lowers the activation energy, which has the overall effect of speeding up the chemical reaction. And so while the substrate or the reactants in a biological reaction are converted into the products, the enzyme itself, once the substrate is released from the enzyme goes back to its natural conformation and can be reused over and over again to catalyze many, many reactions. And this is a figure from your book, figure 5.5, showing the importance of an enzyme's shape in determining its activity as a catalyst. And so this is meant to be the tertiary structure of an enzyme. This pocket is the active site, and then as you can see, the pocket is, or the active site, is the exact shape to bind the reactant or the substrate. Okay, so this is just a picture showing how an enzyme binds its substrate in the active site. So these are the two different reactants, and then catalyzes the reaction and is recycled to its original conformation once the product is released. Okay, so one important concept that I would like for you to take away from this chapter that will help with the next two chapters is that catalyzed reactions in biological systems often occur in a series or in a sequence that together. And so where the product of the first reaction will be the substrate or the reactants for the next reaction until the final product is made. And so when you have a series of reactions that goes together like that, it's called a biochemical pathway. And the next two chapters after this, chapter 6 and chapter 7, are going to talk about two biochemical pathways that you're probably all at least somewhat familiar with: photosynthesis and cellular respiration. Okay, so since enzymes are proteins and their function is susceptible to the same types of environmental factors that proteins are, and we talked about, when we talked about macromolecules, we talked about how temperature and pH can affect the tertiary structure and therefore the function of proteins. Well, since enzymes are proteins, they're susceptible to extremes of temperature and pH. All enzymes have a particular temperature range that they operate best in. If it gets too hot, then the tertiary structure of the enzyme will be lost and it's considered to be denatured or inactive. Same goes for pH. Enzymes function within an ideal pH range. Usually it's human enzymes are between the pHs of 6 and 8. And there are some exceptions to that. Stomach enzymes, for one, usually work best in a more acidic range. Okay, so cells can control enzyme activity by altering the enzyme's shape. This is a term that I would like for you to be familiar with. Allosteric enzymes are affected by the binding of signal molecules, and usually these signal molecules are bound either in the active site or in another place on the enzyme, but the overall effect is that the enzyme's activity is either activated by the binding of a signal molecule, or it can also be repressed or stopped by the binding of a signal molecule. So some signals act as repressors that keep the enzyme from working. Other signals act as activators, which make the enzyme's shape to where it can bind the substrate or the reactant. Okay, so this is just a picture from your book showing how allosteric enzymes work. And so at the top you have an enzyme that is susceptible to repression. And so this is the active site down here. This is the allosteric site. And without having to have any kind of activator signal, this enzyme is active, it binds the substrate, catalyzes the reaction, and the products are created. However, it is susceptible to repression, and so this allosteric site can be bound by the repressor, which changes the shape of the active site so that it no longer is the right shape to bind its substrate. So it makes this reaction, this enzyme, inactive, unable to catalyze the reaction. This one on the bottom is an enzyme that requires an activator signal in order to make the active site the right shape to bind the substrate and catalyze the reaction. So if you look at this first picture, the enzyme is not active. This is the active site. It's not the right shape for the substrate. This is the allosteric site up here. But then if the enzyme binds the activator signal in its allosteric site, it changes the shape of the active site to become the correct shape to bind the substrate, and then the enzyme becomes active and can catalyze its reaction. Okay, so feedback inhibition is a way that cells can control the activity of enzymes where the product of the reaction is a repressor for the enzyme that catalyzes the reaction. And there are two types. There's competitive inhibition and there's non-competitive inhibition. In competitive inhibition, the inhibitor competes with the substrate for the active site. So the enzyme blocks the active site so that it can't bind its substrate. In non-competitive inhibition, the inhibitor binds to the allosteric site and changes the shape of the active site so that no substrate can bind. So competitive inhibition is where the inhibitor is directly competing for the active site. Non-competitive inhibition is where the inhibitor is binding the allosteric site. And this is a picture showing that. So this is competitive inhibition. This is the inhibitor binding the active site, blocking the substrate. This is non-competitive inhibition. The inhibitor is binding the allosteric site, changing the shape of the active site, which prevents the enzyme from binding its substrate and catalyzing the reaction. Okay, so we're going to talk about ATP, and ATP is what is known as the energy currency of the cell. We talked about this a little bit when we talked about macromolecules. Energy from the sun or from food sources has to be converted into a form that cells can use, and that is ATP, also known as adenosine triphosphate. Okay, so ATP powers the cell's metabolism, and there are two processes which we'll look at next that convert energy from the sun and from food into ATP that the cell can use to power its activities. The first one is photosynthesis, and that is where cells are able to convert the energy in sunlight to ATP and then use that in order to make sugars that store energy for the cell. And then cellular respiration is where cells are able to break down the energy in sugars and convert that into ATP for the cell to use to power its metabolism. So that's all I have to say about energy flow in cells and systems. And please look for lectures on chapters 6 and 7 next. Chapter 6 will be photosynthesis and chapter 7 will be cellular respiration. I'll try to have those up by the end of the week, and thank you so much.